Hello everyone, Christy here from Master One Thing. Today we have the honor to have one of the world's leading mTOR researchers, Joan Mannick, as our guest in the podcast. Joan has a background in academic medicine and is currently CEO at her own company, Tornado Therapeutics, where they develop the next generation of mTOR inhibitors. Their goal is to develop an mTOR inhibitor which is better than rapamycin. Throughout the years, Joan has made important discoveries in human when it comes to the connection between mTOR inhibition and the improvement of the aging immune system. Without this research in the longevity field, we will not be where we are today. So in today's podcast, we are going to talk about her research, drug development, mTOR, rapalogs, rapamycin, and other interesting topics. So, Joan, welcome to the show.
It's great to be here. These are all my favorite topics.
Great to hear.
Yes.
One good place to start this conversation is to go back in time when you worked as a physician. What was it that made you interested in the longevity field?
Yes, so I was in academic medicine. I ran a basic science lab and saw patients. And while I was in my basic science lab, I was reading Nature, I think, and there was a review article by Cynthia Kenyon who talked about the fact by mutating one gene in a worm, she could dramatically increase the lifespan. And she said, she put this into perspective that really sort of startled me, saying, this shows that organisms have the capacity to live much longer than they normally do. And that was such a, as a physician, sort of an astonishing comment to go, wait, I am trying to keep patients alive for another week or another month when they have these incredibly serious diseases and just get them through it. But the idea that there may be interventions that could have people live decades longer, not, a month or two longer, was remarkable. And so that got me really interested, and it sort of was turning current medicine on its head, because none of us thought we could really make humans in any way live much longer than they normally do. And that's what first got me interested.
Yeah. How did you succeed in converting this interest to a job in the field?
So I was recruited to a unit of Novartis called the New Indications Discovery Unit. And your job in that unit was to find areas of medicine that fell between the cracks of traditional biotech and pharma silos. Like, it isn't cardiac, it isn't GI, it isn't renal, it isn't endocrine. It's like in between. And so, since reading that Cynthia Kenyon paper, I was really interested in this whole science and like the implications of this science. So I proposed to my boss, I want to study aging, because if we can target aging biology, this could really transform the practice of medicine if we could actually target it in humans. And the best validated way to do this right now is by targeting mTOR, which we can talk more about. And Novartis happened to have a marketed mTOR inhibitor we could use to start looking at this. And I've told this story before. I had a wonderful boss, and he goes, Joan, I totally get why you're doing this, but everyone's going to think you're wacky. So you have to come up with a different idea. But at the time, the CEO of Novartis, Dan Vasella, and the head of research, Mark Fishman, separately had decided, hey, wait a second, the aging field has gotten to the point that we actually might be able to translate this sort of amazing science that they're seeing in preclinical species to humans. So from above, it came down that they were interested in aging. And from my little position, I said I was interested in aging. And so I was given the green light. Go ahead, Joan, go try, see what you can do with our mTOR inhibitors. It's interesting. My first performance review was, Joan's really nice, but she picks terrible topics to work on, like aging. So there was a lot of skeptics when I first started on this path.
Yeah, it must have been hard in the beginning, also when there are not so many people working in the
Yeah,
aging field. But
yeah,
it has improved a lot.
yeah.
Yeah, you touched a little bit on the mTOR. Can you explain what mTOR is, and what is that makes it unique when it comes to longevity?
Yeah, so mTOR is a protein kinase. So it phosphorylates other proteins, but it's evolutionarily conserved. So every species, yeast, worms, flies, mice, all have this protein. And the reason it's so important is it is what links availability of nutrients to growth and reproduction. So when we eat, mTOR is activated, and this allows us to grow and reproduce and cells to divide. And then when we stop eating, mTOR is inhibited when nutrient levels go down. And that's also really important because when mTOR is inhibited, protective pathways are upregulated. So mTOR should have this circadian rhythm on during periods of the day when you eat, off during periods of the day when you fast. And one of the reasons fasting or intermittent fasting may have health benefits is because it decreases mTOR levels and allows these protective pathways to get upregulated. But you really probably need mTOR on more during periods of growth and reproduction. But as we get older and we're not growing or reproducing, we may not need as much. But we need more of those protective pathways that remove damaged proteins and organelles and decrease inflammation. We need more of that mTOR inhibition biology to occur. But actually, what happens as we age is that mTOR, if anything, gets hyperactive and stops responding to fasting. So it's too much, and it's not going down as it should at least in mice, during periods when they're not eating. And in humans, it's been shown it's too high. The levels in human muscle, for instance, are higher than the levels in old people than in young people. So turning down mTOR to young levels or just intermittently turning it off so that these protective pathways gets upregulated is probably good for aging.
Mm, mm. Do we know what specific things that the mTOR targets that may be the longevity benefits?
Yeah, so we don't know exactly, but there's probably multiple mechanisms, protective mechanisms that are good. So for aging. So one is you induce this process called autophagy, which is like a cellular vacuum cleaner. It gets rid of misfolded proteins, dysfunctional organelles, and kind of cleans up the cell. Two, mTOR inhibition tends to decrease inflammation, and there's what's called inflammaging. There's a gradual increase in sort of just low-grade inflammation as we age, which contributes to organ dysfunction, and that's suppressed by mTOR inhibition. Some of that comes from cells that are called senescent cells, and mTOR inhibitors stop the secretion of inflammatory cytokines by senescent cells. There's evidence that mTOR inhibition helps stem cell function. And as we get older, our stem cells, which sort of repopulate our organs with new cells, they become dysfunctional, but mTOR inhibition helps them stay healthy. And then last, mTOR sort of shuts or suppresses the production of new proteins. And as we get older, our cells are like factories making proteins. But as we get older, we have a harder time kind of managing quality control of all this protein production. So by just sort of calming the system down and turning, the factory production down a little bit, it's easier for our cells as we get older to maintain quality control.
Mm, mm. There are, as you said, lots of good longevity data in multiple species. Do you think that the data will also be translated to humans?
This is where I feel really strongly. There is no better validated single drug than rapamycin for extending health span and lifespan. And it's because it's not one lab, it's multiple labs. And every species studied to date, yeast, worms, flies, and mice, show the same thing over and over. These preclinical species stay healthier longer and live longer. And it's really rare to see one compound in all of these different species keep doing the same thing. And because those species span 1 billion years of evolution, it's incredibly unlikely that rapamycin or one of its derivatives is not going to extend something about health span in humans, because the conservation of this effect across 1 billion years of evolution, we have not diverged that much. So it's going to do something. Now, what it's going to do has to be determined. But if I were going to bet, and the other thing is, if you genetically just reproduce the same thing, just inhibit mTOR genetically, same exact effect. So inhibiting mTOR, and particularly mTOR in a complex called TORC1, is so validated that it is impossible to me to conceive that we are not going to be able to find health span benefits of this mechanism of action in humans.
Yeah, it's a super interesting topic. And if we also look at Cynthia's work with IGF-1, that is something that also has an effect on mTOR in
Right.
different ways. So, and also like protein restriction and things like that, which
That's right.
has shown good effects. So there are many things pointing in that direction. So
Right.
yeah, super interesting. Have you talked to Cynthia anything about the connection between IGF and mTOR?
Yeah. Oh, yeah. You know, we're downstream of that gene that when Cynthia mutates, it has this dramatic effect on lifespan in worms. So it's all, you're exactly right. It's all connected to the same nutrient sensing, insulin metabolic pathways.
Yeah, super interesting. You touched a little bit on rapamycin. Can you tell the listeners what that is and how it was discovered?
Sure. So there's a volcano on a remote island called Easter Island. And this volcano has a lake in its crater. And the native inhabitants of Easter Island had said there's something about the water in this volcanic crater that has therapeutic benefits. But researchers went to this volcanic crater and got samples of soil and found it had a bacteria that produced a molecule subsequently called rapamycin, because Rapa Nui is another name for Easter Island. And they found that this molecule produced by this bacteria on this remote island made an incredibly potent and specific, was a incredibly potent and specific inhibitor of this mTOR protein kinase. Then after they discovered this, they discovered all the effects of inhibiting mTOR with this one molecule in all these different species.
What is the weak points when it comes to rapamycin? Does there exist any?
Yeah, so rapamycin and its derivative. So there's rapamycin is approved for use in organ transplant patients because you need mTOR for your immune cells to divide. And when you get an organ from another donor, your immune system sees it as foreign and starts getting activated and replicating and rejecting that organ. So using an mTOR inhibitor like rapamycin stops those immune cells from dividing so they can't reject the organ. So mTOR inhibitors are approved for organ rejection, and they also stop cancer cells from proliferating because you need mTOR for cells to grow and divide. And for those indications, you want to completely shut down mTOR so that there's no cell growth. And when you completely shut down mTOR, there's a fair amount of side effects that patients get. They can lower their immune cell counts. They get mouth sores called stomatitis that are like canker sores. They can get diarrhea. They can even sometimes get a side effect of inflammation of their lungs. So there's a fair amount of side effects. The current mTOR inhibitors or the current rapalogs like rapamycin and its other approved derivatives, Novartis has one called Everolimus, inhibit mTOR both within a complex called TORC1 and less potently, but still partially inhibit in a complex called TORC2. A lot of those side effects may be mediated by inhibiting mTOR in that TORC2 complex. So development of a rapamycin derivative that only inhibits mTOR in the TORC1 complex is predicted to give you a safer molecule that won't have the side effects, or at least as many side effects as you can see with rapamycin as you push doses higher.
You touched a little bit on the name Rapalog. Can you explain what that is?
Yeah, a rapalog is just an analog of rapamycin. So all of these compounds take rapamycin and make chemical variations of that parent compound that sometimes increase bioavailability. That's Novartis's compound. Or there's another one called temsirolimus where they make a prodrug of rapamycin that then turns into rapamycin once it gets into the bloodstream. So, but they all function like rapamycin and inhibit mTOR.
You talked about that mTOR2 inhibition can lead to detrimental effects or side effects. Is there any ways to detect the early signs of mTOR2 inhibition?
So mTOR signals in two multi-protein complexes. One is called TORC1 and the other is called TORC2. And they phosphorylate different downstream targets. If you mutate other proteins that are in this multi-protein complex of TORC1 or TORC2, or you mutate the proteins downstream of TORC1 or TORC2, what you can see is inhibiting any part of that TORC1 complex or downstream targets is associated with lifespan extension. Inhibiting, in contrast, TORC2 genetically actually decreases lifespan in mice and causes hyperlipidemia and hyperglycemia. So in humans, you can look at lipids and triglycerides in the blood, and you can when those go up, that is what we think is a sign that you're inhibiting TORC2, because that's a TORC2 side effect.
Super interesting. Yeah, and you founded the company Tornado Therapeutics, and one of the goals that you have is to develop an mTOR inhibitor, which is better than rapamycin. Could you talk a little bit about that?
Sure. So when I was at Novartis, we realized there's no better validated molecule than rapamycin for extending health span. But rapamycin was coming off patent. All the rapalogs that are currently approved are coming off patent. So there was no patent runway available to develop the current rapalogs for aging-related conditions. So Novartis realized this and put together a large group, and I was part of this group, and most importantly, their expert rapalog chemists were part of this group, and said, can we come up with new rapalogs that are differentiated and better than the first generation rapalogs, and that would have a patent runway that would enable us to be able to develop them for age and test them in aging-related conditions. And this was a very tough task because so many rapalog structures have already been disclosed in previous patent applications. So it took years and years and years of work to get a new portfolio of something really good. And after all these years of work, they came up with a beautiful portfolio of compounds. And most importantly, the majority of these rapalogs only inhibit mTOR in the TORC2 complex. They don't inhibit mTOR in the TORC1 complex. I'm sorry, they don't inhibit mTOR in the TORC2 complex. That's the holy grail we've always wanted of a rapalog phenotype that we think would be ideal for targeting aging biology and safer. By the time this group at Novartis developed all these rapalogs, I had left, a lot of the people focusing on aging biology had left, and this program was orphaned because Novartis was no longer focused on aging. They had just shifted their strategic priorities. So this program became available for in-licensing, and Tornado grabbed it. Like, this is the dream for someone like me. This is the dream portfolio of compounds to see if we can here and now do something to target aging biology and increase health span in humans. Like, this is it. So we're really excited about this portfolio.
Yeah, it seems like you're having a big gold mine in the company.
That's what it's, of course, this is my baby, I'm biased, but I've done other companies. This is the most exciting thing I've ever had an opportunity to work on. So, I just hope we'll get this across the finish line and really make a difference and extend health spans. If I had to bet, this will be our lead compound will be the first drug approved to extend health span in humans, because there isn't anything better validated than this. So fingers crossed, we do what we need to do.
If we go back to 2010, around that period of time, you took a courage step and proposed quite a unique trial to the board. What was it that you proposed?
So when you give old mice an mTOR inhibitor, there are certain organ systems whose function improves, and that's contributes to why they live longer. So it's not all organ systems, but heart function, multiple groups have shown that improves, neurologic function improves, immune function improves, and the animals move better. They spontaneously walk more, and they can, have some measures of better physical function. We decided if we have to just look in a short period of time to see if our Novartis rapalog called Everolimus does something for human aging. It's too hard to look at cognitive function. You have to treat for 1 or 2 years. Heart function also is tricky. Physical function, tricky. Immune function, that's pretty easy to monitor. And it's an organ system whose function we know from vaccines, you can change in a really relatively short period of time. So when you start a program, you want to start with a clinical trial with not a lot of money and not a lot of time, you can get a go, no go decision. So you don't waste money on things that aren't going to work. So we thought, let's just see, can we make the aging immune system work better with this mTOR inhibitor, this Rapalog, and to see if we make the aging immune system better, can we just see if people respond better to a flu vaccination after they've gotten 6 weeks of an mTOR inhibitor? So that was the study. It would be the first study to see, can you make the function of an aging organ system work better in a human when you target this fundamental aging biology pathway?
And the interesting thing here is also that which makes it a courage step is that because mTOR inhibitors are connected to immune suppression, and now you want to test that if they improve the immune system. That feels a little bit odd. I guess that the board also felt like that.
Yes. I got called into a room with all the senior people going, seriously, you're going to use the drug we sell to suppress the immune function to see if it enhances its immune function. No, I mean, like, I think a lot of people thought I was out of my mind. So I explained this is the aging immune system may respond very differently than a young immune system. And two, I was going to use very unusual doses of mTOR inhibitors that turn down mTOR, not turn it off, which is what causes immunosuppression. And I think it was such a weird proposal that if it hadn't been for the senior management of Novartis blessing this idea, I would have just gotten thrown out of the room, because it was a weird proposal given the fact that this drug was approved for immunosuppression.
If I remember, there existed also animal studies which showed those
Right.
benefits. Can you talk also about that?
So really important point. There was a great paper that had been published showing if you treat old mice for 6 weeks with rapamycin and then give them a flu vaccine, they have a much better response to the flu vaccine. It was that study that informed us on how to test in humans. We just took that study and did the same study in humans. Now, they used full doses of rapamycin, which in humans would have immunosuppressed. So the only thing we did differently was we just used very low or intermittent doses of our rapamycin derivative.
How did you come up with the dosing?
So, the great thing about these big pharma companies is they have armies of people to help you. So we had this great modeling and simulation group that could model what dose do you need to turn down mTOR and not turn it off and avoid immunosuppression. And I also had, all these experts who had worked with this, this is again one of their big compounds of Everolimus for Novartis. And so there was people who knew so much about this compound, and there'd been a history. It had been dosed in cancer patients and transplant patients and different dosing regimens. And so we really knew how once weekly dosing, daily dosing, what level of daily dosing led to what concentrations in humans and how that was predicted to inhibit mTOR based on IC50s. Great team came up with the doses.
Yeah, super great data to back things up there. And can you talk about the first trial that you set up and what the results were?
So we took 3 different doses of Novartis's Rapalog, a low daily dose, or 2 different, a low and a higher weekly dose. And these doses were predicted to inhibit mTOR between 40 and 80% during the dosing intervals. So just turning it down partially or more, the more majority of it off. And we treated, and again, part of the reason we wanted to use low doses was or intermittent dosing was not just because we wanted to just turn down mTOR, but also we wanted to keep people safe. And again, everyone thought, oh, you can't use an mTOR inhibitor for aging because it has all these side effects in transplant oncology patients. So the scariest part for me was we have to keep these people safe. Like, do no harm is your number one priority as a physician. So carefully choosing these doses that should be safe was the critical part of this study. But I will tell you, every day in that study, I was just, very nervous, like that no harm is going to get done. No harm's going to get done. Monitoring, just making sure everybody was fine. That was the scariest part of it. Not is it going to work, but just don't do no harm. And I knew if these low doses were doing harm, there was no path forward for an mTOR inhibitor in humans. It's just, there wasn't going to be one. The safety is critical. But fortunately, all the doses were well tolerated. That was in some ways the biggest important finding in that study is you can target this fundamental aging biology pathway in humans safely, which had been really stopped people because they thought it wasn't possible. So we dosed everybody for 6 weeks. It was all fine. Then they had a 2-week break, and then they were given a flu vaccine. And then 4 weeks later, we measured their antibody titers to flu, just like you do with COVID. And the people who got either the low daily dose or the once weekly lower dose significantly improved their response to flu vaccination. And the dose that had the 80% mTOR inhibition didn't work. So that was too much mTOR inhibition.
What age group did you have in the study?
Age 65 and above.
And did you see any difference between age groups?
This only had 50 patients per group, and there weren't enough who were like over 80 to be able to power to say anything about the older age group. In subsequent trials, when we've had higher numbers of older people in bigger trials, if anything, the treatment effect gets better. The older you get, the more benefit you get from the mTOR inhibitor when it comes to immune function.
That is also one question I think some people may wondering about is that, yeah, maybe they think like the immune system is boring or something when it comes to longevity, but it is quite important thing to have a good immune system. Can you talk why that is important when it comes to longevity?
So there was a paper I loved published in Nature from Laura Niederhofer's group. And she's aged individual organ systems and look at the effects of just aging the heart, just aging the liver, just aging the immune system. And she does this by making mice that have a DNA repair defect in different organ systems that just phenocopies aging in that organ system. What she found is if she ages just the immune system, all the other organ systems start aging. And what this suggests is aging of the immune system may drive aging of all the other organ systems. And it kind of makes sense because immune cells infiltrate everywhere. Like your liver stays your liver. And yes, it can secrete some cytokines that may influence distal organs, but the immune system, the cells itself, not only are secreting all these cytokines that's going to influence, but they actually infiltrate into the organ systems. They clean up the senescent cells that are secreting the cytokines. So when you don't have your immune system, you're going to have more senescent cells. They cause neuroinflammation, So I think we've kind of missed targeting We target aging, the dysfunction of the aging heart and heart failure, the aging bone and osteoporosis, the aging brain and Alzheimer's, but we haven't been focused on targeting the aging immune system. And I bet you there's all sorts of interesting therapeutic effects if we can do this successfully.
Yeah. And I think, is it not also a good immune system also can prevent cancer and things like that?
Absolutely.
Yeah.
Now all the cancer immunotherapies are pointing to just this. You make your immune system work better, you can fight cancer better because the immune system can get rid of the cancer cells. But it's probably much more than just cancer. It's probably, I'm guessing Alzheimer's, kidney, lung, all these things, if we can reset the immune system, not everything. I mean, nothing is good for everything, but I bet you we'll have new therapeutic plays on beyond just autoimmune disease, on different pathologies.
And if we look at the influenza, for example, some people may think, okay, influenza is not so dangerous. What would you say to them?
Wait till you're 75. It's like there's really interesting papers where you look at when over the year people die, and people under 65, under 75 really die this about the same level, whether it's summer, winter, spring, fall. When you get to 75 and above, there's these peaks of death around January of each year, corresponding to winter cold and flu season. And it's because not only do the respiratory viruses kill via pneumonia because your immune system isn't as good, but also, even if you don't get the pneumonia, they exacerbate underlying comorbidities. And there's a paper in New England Journal showing even after a common cold, the risk of having a heart attack or a stroke, a cardiovascular event, triples for 2 weeks, triples after a cold. Now, that tripling isn't occurring in young, healthy people. It's occurring probably in people who have underlying cardiovascular disease. Whether that's tripling is because there's an exacerbation of inflammation by the virus, which then exacerbates your underlying atherosclerosis, or other reasons. But it's known, and you could see it in COVID. Even if that old person didn't die from COVID itself, there was all these sort of like domino effects of other health issues that happen in older people get COVID that will kill them. So being able to control these respiratory tract infections could have a huge, by getting your immune system better able to fight them, could have a major impact on morbidity and mortality in older people.
Really great answer there. And you did also a phase 2 trial. Can you talk what you looked at there?
So we did 2 trials at Novartis looking at vaccination as a primary endpoint to say, if you give people an mTOR inhibitor, do they respond better to a vaccination? In both trials, we showed they did. In the first trial, we noticed that everyone who got the lower doses of mTOR inhibitors were reporting having fewer respiratory tract infection as adverse events. So when you do a clinical trial, people have to report anything bad that happens. I had a headache, I fell, I had a runny nose. They're required to report everything so that you can see if your drug has a side effect. So what they were reporting is that people at placebo were saying, I have had a respiratory tract infection more often than the people who got an mTOR inhibitor. So we thought, and these weren't flu. It was like all-comer respiratory tract infection. So we thought, maybe we're making the immune system respond better, not just to a flu vaccine, but to all sorts of different infectious pathogens. So in the second trial, we asked everybody to report anytime they had any kind of infection. And we compulsively collected this by having them fill out infection diaries, not just report adverse events when they came for a clinic visit. And we had the sites call and ask them, have they had an infection? In that trial, we found that people who got low doses of mTOR inhibitors reported having fewer total infections, and that persisted for an entire year after they were dosed with just 6 weeks. But most of those infections, think about it, when you get it, what is the most common infection you get? It's a cold or a bronchitis. It's always a respiratory tract infection. So most of those infections were respiratory tract infections, and people were having significantly fewer respiratory tract infections again. We then, as part of this trial, did what's called non-hypothesis driven RNA-seq. We said, let's just take the blood from the people who've got an mTOR inhibitor and just see what gene expression pathways change in older humans targeting this fundamental aging pathway. And we thought, oh, we're going to see increased autophagy or better mitochondrial function, all these things linked to a healthy lifespan. But instead, almost every pathway that was significantly changed, and it was always significantly upregulated, were antiviral pathways. These are the pathways that are deficient in older adults. One of the reasons they get more severe infections are what's called interferon-induced antiviral pathways. So what it looked like is what we're doing is upregulating antiviral immunity in older adults, which is deficient. So we're making the organ system work better, and we're protecting them from getting respiratory tract infections, which are almost all viral. So from that phase 2 trial, that was our take-home message. And in that trial, we used 2 different kinds of mTOR inhibitors. We used a rapamycin derivative, a Rapalog, and we also used a catalytic site inhibitor. It inhibits mTOR a different way, and they both worked. But since the Rapalog was coming off patent, we moved forward with a catalytic inhibitor because it had enough patent life for us to be able to develop it for improving antiviral immunity in older adults.
And the new type of mTOR inhibitor, I think you called it RTB101.
Yep. So it was a drug at Novartis that was at Novartis called BEZ-235. We then spun it out into its own company called Restore Bio, and the name changed to RTB101. It wasn't a Rapalog, which is the most validated class of therapeutic. Again, it targeted the same thing, but it was a little different. But it was at the time, that portfolio of Rapalogs wasn't ready to be used in humans. So this was the only mTOR inhibitor available that we could move forward at that time for targeting aging biology. So we did 2 large phase 2b and phase 3 trials with this RTB101. The phase 2b worked. We decreased the incidence of viral respiratory tract infections in high-risk older adults, and the phase 3 failed. But in both trials, and the phase 3, part of the reason it failed is it turned out it was underpowered because we had much lower incidence of viral respiratory tract infections in the phase 2b and phase 3. But in both trials, the lessons learned from those trials was we probably hadn't had the right endpoint in either trial, because what it looks like is in both trials, these antiviral genes were upregulated, but they seem to have a bigger effect on decreasing severity than incidence of infection. And where they really seemed to have benefit was in people 75 and older, not the 65 to 75. So I think there were lessons learned. It was a very painful lessons learned to have a failed phase 3 trial. But I think it's important in these failed trials to look at the data and go, were we just wrong? mTOR inhibitors don't help the aging immune system? Or was the trial wrong? And we weren't able to show the effect because the trial design wasn't right. And I think looking at all the data, it's pointing to the trial design wasn't right. And we should move forward with a different trial design should we decide to move forward in this area.
And one thing that I have heard from people who seek simple answers. They tell, for example, oh, look at the phase 3 trial. It failed to meet the primary endpoint. That means that mTOR inhibitors don't work. But it's not that easy.
Not at all.
When in research, you do experiments. If every experiment will work, then then we don't need to do experiments.
Right. When you move into a new area, it's always high risk. Drug development, period, is high risk. And you have to just do the best experiment you can think of, given the data you have at hand at the time, and learn if it doesn't work, why, and get smarter and better. And that's how we're going to get this field forward. Absolutely. This does not tell us that mTOR inhibitors aren't going to help aging. I think what we've never published is we did a subsequent trial after that failed phase 3 where we said, let's just look at really old people and look at a severity endpoint. And we did a trial of preventing severe COVID-19 in residents of nursing homes experiencing COVID-19 outbreaks. And this was before there were any vaccines available. And what we found is that people who got the same mTOR inhibitor had no severe COVID-19, whereas 23% of the people who got placebo got severe COVID-19 and half died. And it was a small trial. But again, this is suggesting that maybe these drugs actually do work. So the jury's out. I think that the predominance of evidence would suggest this is still worth pursuing, but we have to do more trials to know.
Yeah, yeah. If we look at the RTB101 and compare it to Everolimus, the dose protocols differ. Can you talk a little bit about that?
Yeah, so for the RTB101, we gave it very low doses once a day. And for Everolimus, you can either go very low doses once a day or higher doses once a week. The reason you can dose Everolimus once a week is it has a prolonged PD effect. It inhibits mTOR for like 3 days if you give a 5 milligram dose once a week. BZ has a shorter half-life and shorter PD effect, so its effect is gone, after 24 hours. So you can't dose it as easily once a week.
That makes me also think on the mice studies. For example, when you give rapamycin to mice, the half time for rapamycin is very low and probably more aligned with RTB101. Is that something you have looked at or think about?
No, it's a really good question that I don't think is thought enough about. Mice handle rapalogs much differently than humans. So in mice, 99.9% of rapalogs are protein bound. Very little of it is free. So your dose that you have to give a mouse to achieve the same level of mTOR inhibitor as what you have to give in a human is very different because in humans, 25% of it is free. So, and humans, rapamycin goes into red cells and sits in red cells, and that isn't true in mice. So the whole pharmacokinetics of rapamycin in a mouse and rapamycin in a human is very different. And I think lots of times when people, they just go, oh, well, if that's the dose in the mouse, that's the dose of the humans. No, you have to do sophisticated modeling and simulation to understand all these differences in how the drug is handled between species to understand how to predict a dose.
If we compare the safety profile with Everolimus, Is RTB101 better or the same, or how is the safety on that?
So RTB101 is a really interesting molecule because it inhibits the catalytic site of this protein. It competes with ATP. And there's other compounds that also are catalytic site mTOR inhibitors. But what's unusual about BEZ or RTB is that it inhibits the catalytic site of mTOR in the TORC1 complex at lower concentrations than it inhibits the catalytic site of mTOR in the TORC2 complex. For unclear reasons, somehow it gets access at lower concentrations to mTOR in the TORC1 complex. So at low doses, it is TORC1 specific. That's not true of a lot of the other mTOR catalytic site inhibitors that are equipotent against TORC1 and TORC2. As you increase the dose of BEZ, it not only will inhibit TORC2, but at even higher concentrations, it will inhibit PI3 kinase. And so it was originally developed to be a dual PI3 kinase mTOR inhibitor for cancer patients. It turns out you can never get high enough doses, or it's really hard to get high enough concentrations in humans to inhibit PI3 kinase. And in cancer, it functioned basically as a TORC1 or sometimes a TORC1/2 inhibitor. So it failed in cancer. But what made it fail in cancer is exactly what you want for aging. But in preclinical species, if you keep pushing that dose, it will have even more tox than rapamycin because it can inhibit not only mTOR but also PI3 kinase. So the more molecules you inhibit, the more toxicity you will have. But at low doses, it was incredibly safe in humans because we were using 1/120th the maximum tolerated dose in humans, which was established in cancer patients. It was tiny. And it had just as lovely side effects as little doses of Everolimus. They both, at low doses, are very nicely tolerated because in general, you're not inhibiting TORC2 with very low doses of either of them.
And if we look at vaccine and mTOR inhibitors, some people may think like, why take mTOR inhibitor? Why not just take a vaccine instead? Because then I will improve my immune system for that influenza virus. But what would you say to those people?
So there are hundreds of different respiratory viruses that are circulating. People always assume, I'm sick, I have COVID or flu. And now they know RSV, but there's parainfluenza, there's adeno, there's metapneumo, there's rhinovirus, which is the cause of the common cold. There are 170 serotypes of rhinovirus. You know how there's like Delta and Omicron and 6 different serotypes of SARS-CoV-2? 170 of rhinovirus. We've never been able to develop a vaccine, never been able to develop a therapeutic. And rhinovirus is the most common cause prior to the COVID-19 pandemic. It was the most common cause of pneumonia requiring hospitalization in people over 80. Rhinovirus, the cause of the common cold, no therapies. So yes, vaccines are really important. They will protect you from a specific virus, but there's so many other viruses out there that can kill you when you get older, that if you could have something that boosted your immune response to all viruses, you would be further protected. And this also might help you boost your response to a vaccine. And that response to vaccines tends to diminish as we get older.
Yeah, very good answer there, because as you say, there are thousands of different mutations out there of viruses, and a vaccine is more specific and mTOR is more broad.
The two together are good. Now, this is provided we prove in a randomized control well-powered trial that the mTOR inhibitor does what we think it's going to do and boost your antiviral immunity and protect you from getting severe infections. That's We have to show that, but if it does, that is a big additional help to older adults to keep them healthy.
And if we look at the dosing, is it better to, let's say, to take the mTOR inhibitor before the cold season and take a pause during the cold season, or is it better to take it all the time? Do you have any data on that?
So all the studies we've done are either with 6 weeks or 16 weeks. Just taking it during winter, like the beginning of winter cold and flu season, to sort of help you boost your antiviral response to the viruses you might get exposed to. You always want to give as little of a drug as possible for as short a time as possible, because every drug that has a biologic effect will have a side effect. So the less you take, the less side effects you're going to have. What was interesting is the 6 weeks in one study, and this has to be repeated, seemed to have a prolonged benefit, as though the immune system was kind of reset and stayed reset. If we keep showing that, it may not be you have to treat the entire winter cold and flu season. You could treat for 6 weeks and then you'll stay with a better immune response. We just don't know. And this is why, you'll ask, do I take rapamycin? No, because we don't know yet what's the right dose, what's the right duration, where is the risk benefit ratio right? All of these questions are answerable and need to get answered in well-controlled, placebo-controlled trials so that everybody gets the maximum benefit with the least risk.
If we look at also when people get, for example, the influenza, is it better to take a break or to continue taking
Yeah.
the mTOR inhibitor?
It's a good question because I can't remember the details. I know the FDA, when they were looking at this, gave us recommendations of if someone gets the flu to continue or not continue just until we know. My guess would be the best thing to do, if we prove this is working and decreasing severity of infection, is to continue to take it to boost that response. But again, you always want to maximize safety and make sure that is correct before exposing people to any risk. So I think the answer is we don't know yet.
No. Is there any good biomarkers to look at when it comes to the immune system?
This is a great question, and I will tell you a lot more work needs to be done. What we've seen in people who get mTOR inhibitors are two things. One, their level of exhausted T cells goes down. So PD-1 positive, CD4 and CD8. That may, when you have less exhausted T cells, they may be able to function better. And we always see these antiviral genes get upregulated. Now, we've looked in populations of 50, 80, 100 people, comparing this upregulation of genes and the people get mTOR inhibitor versus placebo. There's enough variation from person to person in your baseline level of these antiviral genes that I don't know on an individual level, are we going to be able to use this as a biomarker? It's something we have to work out.
If we look, for example, at the ITP, then many different experiments that they have done there show gender-specific effects. Have you seen also gender-specific effects?
We don't in humans so far. The males and females respond the same.
Okay. So what other interesting areas are you looking at than just improved immune system?
I think sarcopenia is a really interesting area. So first of all, you can use rapalogs for non-aging related diseases. And I'm not going to talk about this because I'm assuming you want to focus on aging.
You have the words, so you can.
So, like rapalogs are approved for cancer, they're approved for transplant. They may be quite good for autoimmune disease. There's cancer indications they haven't been tested in because they were coming off patent. They may be, there's all sorts of things that where there'd be of potential use. But for aging related, because I think that's targeting the aging biology and targeting some of these areas of medicine that we haven't tapped into at all, like the failing aging immune system, like our failing muscles. We have nothing for sarcopenia, and people are testing things, which is great because sarcopenia is a terrible impact on quality of life. When you're getting too frail to go and get on an airplane and walk through an airport and take your dog for a walk and visit your grandkids. I mean, it's a terrible and nothing. We have nothing except some resistance training and higher protein intake, which people can't always comply with. There's some really, really interesting data with rapamycin that it may help not build muscle because you need mTOR to make proteins and lipids. But even though it doesn't make muscle bigger, it seems to make muscle in older mice and somewhat also in older rats function better and be stronger.
Mm.
So Novartis had a wonderful program to build muscle for drugs that make muscle bigger, but it turns out even if you gave sarcopenic people this drug that made their muscle bigger, they didn't get any stronger. So it's not about the muscle size, it's about the muscle function and strength. And mTOR inhibition may be a really interesting way to make old muscles function better.
Is RTB101 your leading candidate in your portfolio? No?
We don't even have that. That drug went back to Novartis. So our whole portfolio is all new rapalogs. I like them better. I loved it's not negative about RTB, but RTB wasn't this validated. It's rapamycin that has been so well validated, and all of my compounds now are rapamycin derivatives. So this is the one where I feel most confident that we should be able to get this to target aging biology just like rapamycin, except our compounds may be even better because they're TORC1 selective.
What was the leading candidate name?
Our lead candidate is called TOR101,
Okay.
and we haven't gotten it into the clinic yet. We're going to have to test phase 1. You do just safety and efficacy and make sure you get exposure in humans and make sure it's safe.
And is it also daily dosing instead of weekly dosing, or how does it?
Probably can do either because it looks very similar in how it behaves to rapamycin or Everolimus, the Novartis drug. They all behave very similarly, and we've tested them in monkeys, which should predict closer than a rat how it's going to behave in a human. What's remarkable about this drug is its safety. So before you go into humans, you have to do 28-day, what's called good laboratory practice, GLP toxicology studies in 2 species. And the 2 species you usually do with a rapalog are rat and monkeys. So you dose for 28 days and you push the dose up to see where do you start seeing toxicity, at what dose, and what are the organ systems where you get toxicity. And you find the dose, the lowest dose with no toxicity to determine what your human doses will be in the clinic, because you want to make sure you're safe. With Everolimus, Novartis's rapamycin derivative, you start seeing toxicity at like 0.25 mg per kg or 0.5 mg per kg in rat. After 28 days, and you see tox in thymus and heart and lung and uterus in testes. Same study with TOR101. We have dialed out all the tox except the testicular tox because you need TORC1 to make sperm. Sperm have to grow and divide, and if you don't have TORC1, they can't be made. But we knocked out the lymphoid toxicity. It's dialed out. Heart toxicity dialed out. Lung toxicity dialed out. Uterine toxicity dialed out. We've pushed the dose all the way up to 250 mg per kg, and in those organ systems, no toxicity. It's remarkable. And yet, when you look at TORC1 inhibition, it's beautifully inhibiting TORC1 in all sorts of different organ systems, but without causing tox. Monkeys, we haven't been able to find any tox up to the highest dose we can dose in a monkey. So this is so exciting because we've got what seems to be a really safe TORC1 specific rapalog. And that gives us the opportunity, to push the dose higher than we could ever push the first generation rapalogs, because if this preclinical tox translates to humans, this benefit, we can we have much more freedom to test different dosing. So it's going to be exciting just to see, how does this compound behave in the clinic? Is it really, really safe? And what can we do with it for aging-related conditions?
Yeah, super interesting thing. And what are the next steps around that compound?
Yeah, so we're going to bring it into the clinic, hopefully first half of next year, test it single ascending and multiple ascending doses in normal volunteers, and look at its pharmacokinetics, its, safety, and see how it looks in a human. But we're optimistic we've got a good one.
Will you look at the improved immune function or
So one of the interesting things you can do with this compound for the antiviral indication is do a trial after you establish safety and exposure in your phase 1, is a trial called a Rhinovirus challenge trial, where you give everyone a virus, and therefore you're not dealing with what we dealt with in the phase 3, where no one got it, or like the number of people got infected is 50% lower than what you thought. Everyone gets the same level, same dose of the same virus at the same time,
Mhm.
and look to see, do people who get an mTOR inhibitor have less severe infections than people who get placebo? And
Mm.
it's not an expensive trial in the scheme. All clinical trials are expensive, but in the scheme of clinical trial cost, it's not that expensive. And it will give us a really nice go, no go decision. Is this an indication we should continue to pursue, or does this look like all that data we were just probably wrong, and mTOR inhibitors, even if they upregulate these antiviral genes, it's not doing anything?
I'm really looking forward for that
Yeah.
trial there. And will you also use vaccine or only mTOR inhibitor or?
So this is the cause of the common cold, the rhinovirus. No vaccines, there's no treatments. You know, it's mTOR or bust. Right now.
And when do you think the results of that trial will be published?
Yeah, it's a good question. As soon as we get the phase 1 done, then we can start that rhinovirus challenge. So it just depends on phase 1 timelines, but hopefully, end of 2024, early 2025, but we'll just have to see.
And I think many people are very curious also about when do you think we will see mTOR inhibitor for humans?
Approved.
Yeah.
I think 2030. I think if we can do an expedited clinical development in one of these indications and it works, and we be efficient and aggressive with timelines, I would think 2030.
And it feels like Tornado Therapeutics is the company who is one of the leading companies in mTOR inhibitors. So most likely your company will be the first one pushing this forward, is my guess.
Yeah, I mean, I think what is our differentiator in particular? There's Aovian is another lovely company with a TORC1 selective Rapalog, but they're focused on rare neuro diseases, as far as I know. I think what makes us a little differentiated is we're really motivated to take this preclinical huge data set showing that benefit for aging related conditions and take advantage of that and push these forward for aging related diseases.
Do you have any plans on doing any lifespan studies on animals?
Yeah, it's a great question. We have a portfolio of these compounds. You don't want to take the compounds you're moving into the clinic and start doing all sorts of new preclinical experiments and mucking the waters. But we have other TORC1 inhibitors that are very similar that I think would be very interesting to test in lifespan extension studies.
And how big is your portfolio? Is it a big one
We
or?
have 80 different rapalogs that we've in-licensed.
Yeah, that's that's a really big one.
Yeah.
Then you have quite much work.
Yes. Yeah.
Yeah. Do you think the ITP would be interested if you sent in a proposal?
I would think so. Yeah. It will tell us, is a TORC1 inhibitor better than a TORC1 2?
Mm. I think there is a possibility every year to send in a proposal, and the deadline is like the end of February. And if they choose that proposal, then it's a free ride, which is quite nice.
Yes.
Yeah, hopefully we can see that in the future. So,
Yeah.
yeah. And you talked a little bit about that you don't take rapamycin. What data do you want to see before you take it?
I would like to see data. If I were going to take a compound, I like this new compound, and granted, I'm biased, but it's so safe and it's hitting the same thing that I want rapamycin to hit, which is TORC1. And then I'd want to see what's the right dose, what's the right duration, and do I have compelling data, for instance, on respiratory tract infections. If I take this dose, I'm not going to likely have any side effects, and I am likely to have less severe respiratory tract infections. That's how you get a drug approved. You get that data set. And I think there's a reason the FDA is around to make sure that we're not taking anything where the data supporting it is insufficient for our safety. So I'm kind of a rule girl, and I like the FDA approval, even though sometimes the FDA can disagree when you're in, drug development. But I think in the end, they're a really, really important safety, gauge in making sure nothing gets to us that isn't really safe and understood.
Mm, mm. If we look at the different longevity interventions, what would you say are the four most promising longevity interventions for humans?
Rapalog here and now, number one. For the future, I think epigenetic reprogramming is just remarkable because, again, you can reverse, not just extend lifespan, you can make things younger. I think it's the safety, getting that safely done is going to take longer. That's why it's not ready in the here and now, whereas the rapalogs are. But I think behind the rapalogs, it's the epigenetic reprogramming is just remarkable science.
And do you have the third and fourth one also?
I think the whole senolytics and senescence, that's a really interesting area, although it's more complicated, I think, than we realize to be able to target a senescent cell, not hit another cell, and each senescent cell might need to be targeted differently. So I think it's complicated, but yet its role in the inflammation that clearly is involved in organ dysfunction is really interesting. I think targeting Anna Maria Cuervo has some beautiful data on chaperone-mediated autophagy, where she has had, beautiful effects in many different organ systems. So figuring out ways to target her pathway is also really interesting.
It's interesting you touch about the autophagy thing there. Have you looked at mTOR inhibitors, if they target autophagy differently?
Yeah, so our compounds, our TORC1 specific compounds, induce autophagy just like rapamycin and Everolimus. I think for mTOR, for rapalogs and mTOR inhibition, it induces autophagy in some cell types, but not, I should say rapalogs induce autophagy in some cell types, but not others. And whether our 80 compounds, some are better autophagy inducers than others, we don't know yet. The catalytic site inhibitors are better at inducing autophagy consistently across cell types. And when you use a catalytic inhibitor plus a rapalog, that's also a very efficient way of inducing autophagy. So it's mTOR inhibitor and cell type dependent.
And as you talked in the beginning there, autophagy can be one of the important mechanisms why the life extension in multiple species are created. So yeah, super interesting. What would you say are the biggest challenges in the longevity field?
I think it's the clinical development pathway of finding there's the current way of, oh, you take a non-aging related rare disease or something and you test your compound and then you get approved there, and then you can take the higher risk to the aging related diseases. But I think that how do you move forward in sarcopenia? How do you move forward in the aging immune system? How do you move forward in these frailty, these sort of really important aging related conditions that no one has gotten a drug approved yet for, and yet are incredibly unmet medical needs? You have to be willing to accept we have to try and fail and understand we may fail and learn from the failures and keep going, because it's an untrodden path. And so it's like what we tried at Restore Bio. We tried, we had a failure, we learned, but we can't get scared by it. And we have to then go, how do we do it better? And it's like Elon Musk, he always says at SpaceX, his rockets blow up, but he said, it's okay. It's okay to blow up because we learn and we're going to do it better and we're going to do it better. And each time it blows up, we've learned something more, and then he gets it. There may be, we have to have a mentality of not like panicking if we don't get the answer we want, but having perseverance and keep iterating and getting it right for these difficult new clinical development paths.
Yeah, it's very important not to give up, and I think I can talk for many of us that I'm very thankful that you didn't give up when you got the phase 3 result, because you saw that something was wrong and you just changed different things and yeah, do new experiments and learn from it. So yeah, one rocket blowed, but you're building the next one here now.
Right. I hope it's not very many blown rockets.
Yeah, good. Is there any help you need in moving the field forward, like funding or something else?
Yes, I think this whole field, what you really need, I could speak for biotech more than academia. And of course, it'd be great if NIA had lots of money to fund all the great projects that people are doing. But for biotech, it is essential to get really good, seasoned institutional investors invested in companies because they give great wisdom, great direction, and people follow them. And we have to be able to get the institutional investors to understand the field and understand the potential of the field and create companies that are speaking their language, um, and developing drugs in a way that they see as sensible, because that validates the whole field. And we're not quite there, we're getting there, but that will be a big help, is to have this sort of an accepted, exciting area for these blue chip institutional investors.
And where can people find you online?
Well, I'm not a big social media presence, but they can find me on LinkedIn, probably LinkedIn, or just reaching out to the company.
I will put up your contact information in the description so that people can find you if they want to collaborate or something else. So
Sure.
yeah, good. I think that's all, and it has been really great to talk to you, Joan, and a big thanks again for all the great work you have done and keep doing.
Thank you so much, Krista. It was a pleasure to be on it.
Yeah. So take care and good luck with everything. Disclaimer. The podcast is for general information and educational purposes only, and is not medical advice for you or others. The use of information and other things linked to the podcast is at the user's own risk. Always consult your physician with anything you do regarding your health or medical conditions.